Good afternoon, everyone, and thank you for joining us today. My name is Katy Stubbs, and I lead the public engagement team here at Alzheimer's Research UK. And I'm delighted to welcome you to the 12th event in our Lab Notes online series. Today, we're hearing from scientists in our Southwest Research Network, who will talk about brain support cells and the growing understanding we have of their role in Alzheimer's disease. But first up, I'll just go over some housekeeping for the Zoom platform that we're in. So during the event today, you are welcome to switch on the automatic subtitles using the CC button at the bottom of your screen. They're not 100% accurate during the live event, but they will be edited so that they are correct on the event recording. And if you've missed previous events, you can watch any of them back in your own time, as they're all available on the Lab Notes webpage or on our YouTube channel. And we'll just pop a link in the chat now. During this event, if you would like to ask a question, you just need to click the Q&A button at the bottom of your screen, which will bring up the Q&A box where you can type your question. Please do submit questions throughout the event, and we will answer as many as we can in the Q&A session that will happen after the talks. So now we're just going to ask you a few poll questions, because while we can't see you due to the online format of this event, we're really keen to know just a little bit more about you. So those should pop up on your screen now. And they just ask you to rate your current knowledge of dementia research, which of the options best describes your reasons for attending the event today, and whether this is your first Lab Notes event. So I'll just pause while you get the chance to pop in your answers on there to give us a good understanding of who we've got with us today. So I can see those answers starting to come in now. Just give it a moment longer and then we'll share those back with you. Wonderful. So most people rating their current knowledge of dementia research around average. Hopefully after today, you'll feel that's improved a little bit. And a mix of reasons why people are attending today. So most people are coming because their work is related to dementia. And then kind of after that, it's you've got a friend or family member with the condition, or you have you don't have a personal connection, but you're just interested in finding out more. And almost an even split on people coming along for their first Lab Notes event. So we're really glad to have you if you've come along for your first one, and also really glad if you're joining us for a kind of a second or third or fourth or maybe even the 12th time. That would be amazing. So thank you so much for coming along today. So before we move to the talks, I just want to share some little bits of news from us here at Alzheimer's Research UK. So today is the 21st of September, which means that it's World Alzheimer's Day, which is an annual global awareness day for all types of dementia, not just Alzheimer's disease. And while as a charity, we fund pioneering medical research, one of the other things we really want to do is improve public understanding of dementia and research. And so as part of that today, we've released findings from our latest Dementia Attitudes Monitor. So I just want to share a few highlights from that with you. So our Dementia Attitudes Monitor report gives a detailed insight into the UK's attitudes towards and understanding of dementia, and it highlights key areas for us to focus on in our work. And this is the second time that we've conducted the monitor, having first done it back in 2018. And this work and the work we did back in 2018 has revealed areas where awareness and attitudes and understanding have changed over the recent years. And so I just wanted to cover some of those findings. So first up, this is similar to what we found the first time, which is that more than half of people in the UK have been affected by dementia, most often a grandparent or parent. And this serves to highlight the impact that dementia has on so many of us. We also found that there is a shifting understanding of dementia with increases in a couple of areas. So we've seen an 8% increase in people correctly disagreeing with the statement, dementia is an inevitable part of getting older, and an 11% increase in people agreeing that dementia is a cause of death. However, hand in hand with that, we've also seen an increase in fear around dementia compared with our 2018 survey. And when we asked people whether they thought it was possible to reduce their risk of different conditions, we found that while 75% of people believe they can influence their risk of heart disease, and that's 78% for diabetes, only 33% thought that they could influence their dementia risk, despite the risk factors being in our control, being quite similar between these different conditions. We also saw a difference between men and women, with women less likely to think that they could affect their risk of dementia. But despite this limited understanding of the ability to reduce dementia risk, when we changed that question, we saw that 75% of people believe that they can influence their brain health, suggesting that positively reframing dementia risk reduction as protecting brain health could be a major opportunity to increase engagement on this topic. And this is work that we've started doing this year already. So you may have seen our Think Brain Health campaign, which we created to help raise awareness of the different brain health factors and some of the simple steps that we can each take to look after our incredible brains. We also found that there is strong support for medical research and the potential breakthroughs on the horizon. So the majority of people that we surveyed have a positive outlook, with 61% of people believing that one day the diseases that cause dementia will be cured. And interestingly, this also translates through into support for getting involved in research, with a big increase of 19% in the number of people who would consider getting involved in research studies. During the pandemic, the shared experience of coming together to help science overcome COVID has given people greater trust in medical research in general. And this is really positive, as willing research participants are vital if we are to deliver breakthroughs for dementia. So if you are interested and you're not already signed up and you want to find out more, then do check out Join Dementia Research, which can help you find ongoing studies that you could be a part of. So these are just some of the findings from the Dementia Attitudes Monitor. So if you're interested in finding out more, do check out the website where you can also find the full report. So the bulk of today's event, we're hearing from researchers in our Southwest Research Network. And this network is one of 15 that we support across the UK, providing funding for research, networking and collaboration opportunities, and also helping researchers to share their discoveries and their progress with the public. So our speakers today will be talking about brain support cells called Glia, and what these cells do in the brain and why we're realizing how important they are in diseases like Alzheimer's. So first up, we'll hear from Dr. Konstantin Glebov from Plymouth University, and then speaking second is Dr. Jon Witton from the University of Exeter. Once they've given their talks, I'll come back on screen and we'll move to the Q&A session. You can submit questions again at any point during the event, and we will try and answer as many as we can in the time that we have. So without further ado, it's my pleasure to introduce our first speaker, Dr. Konstantin Glebov. Over to you, Konstantin.
Thank you, Katie, for great introduction, and it's great to be here. Welcome, everyone. Let me just switch to my slides. So I hope you can see it now. There is no doubt that we have made astonishing advances in understanding how our brain is functioning, and from a concept of brain working as a cooling device to description of major cellular components like neurons and glia cells to complex networks we are able actually to image and analyze today. And while significant resources have been dedicated for study of one particular type of brain cells, neurons, and most of you are familiar with, today my colleague Jon and I are going to talk about the other side of the brain and introduce glia cells and their role in Alzheimer's disease. And from the first case that was described more than 100 years ago, central dogma of research into Alzheimer's disease promoted a neuron-centric approach. And this should not come as a surprise, since prominent neuron-specific phenotypes were observed in patients and disease models, and included loss of neurons and synapses, which are interneuronal connections, as well as observation of intracellular aggregates in neurons. At the same time, observed changes in glia cells believed to be just a reactive response to neuronal damage. And with finding of AD-related mutations in proteins like gamma secretase or APP or ApoE in the early 90s, further solidified this preconception for several decades to come. Our understanding of glia function is constantly evolving. However, initially we believed that glia cells served as a mere glue, the term coined by great scientist and clinician Rudolf Virchow, and this word glia finding its origin in Greek word for glue. So this glue is supposed to hold fragile neurons together and provide some sort of scaffold for them to exist and function. With time, we started to see them more as neuronal helpers that help to provide neurons with nutrients and perform other supportive functions like fighting the diseases and providing guidance for neurons to grow. And finally, with better understanding of critical role of glia in brain functioning, and in certain circumstances, we actually can see glia cells to some degree as neuron overlords and their masters, since now we know that glia cells can control and direct key aspects of neuron biology and development. And of course, there is a degree of oversimplification in all of this. But because of the neuron-centric focus, one can say that glia cells were somewhat neglected by researchers as well as funders. We can see that glia represents only 38% of Google results in comparison to neurons in connection to Alzheimer disease. And this should come not as a surprise. This should come as a surprise, actually, since glia cells outnumber neurons by a significant margin, and the data is varying, but it is around 50 to 1. Glia cells and their functions been extremely diverse, and if simplifying things just for the sake of this event, then one can split glia cells into 3 subtypes, with each variety specializing in certain tasks. For example, oligodendrocytes help neurons to communicate faster by providing myelin insulation around neuronal long processes called exomes. Astrocytes, on the other hand, involved in the blood-brain barrier and provision of nutrients and have a role in the repair and scarring processes following infection and traumatic injuries. And with the last type of microglia, or brain resident macrophage or immune cell, and they basically considered to be a brain police. Main function of microglia is patrolling the brain for signs of infection or disease, and their interesting ability, both biologically and clinically, is that microglia can eat things up, as well as produce pro and anti-inflammatory substances. They're also important in controlling number of unnecessary synapses in a process called synaptic pruning, where microglia approaches synaptic connections, the connection between 2 neurons, and basically eats this connection up. So how this is relevant for Alzheimer's disease? We can see that microglia cells are critically involved in numerous processes, from controlling number of synapses to clearing debris and pathogens, including protein aggregates like amyloid plaques, as well as being involved in controlling neuronal death. During Alzheimer's disease, one can detect multiple changes in glia and microglia cells in particular. And we can basically separate microglia functions into 2 categories: loss and gain of function. Loss of function is when microglia cells actually are not able to perform as usual, thereby limiting cells' responses to a disease-related changes in the environment. On the other hand, gain of function can be seen as something when cell is overly enthusiastic, and what they're doing actually, they're thereby doing more harm than good. In Alzheimer's disease, we can see actually that both loss and gain of function are detectable, and it depends either on the glia or on a particular pathway that is affected in the glia cell. Findings about TREM2 receptor that were made actually less than a decade ago triggered a major paradigm shift in the field, as we were able to propose a more central role for microglia in Alzheimer's mechanism, thereby displacing neurocentric view and providing a more complex and balanced theory. And this paradigm shift sparked an explosive interest in microglia biology, as a crucial role of the immune system in dementia was taking hold in AD research. Discovery of TREM was a milestone discovery in Alzheimer's disease research. And now, after a decade of work, we can understand how changes in microglia behavior and intracellular processes can contribute to Alzheimer's disease. But of course, we still got a long way ahead of us until we can fully understand the role of TREM2 in microglia functioning and how we can use this knowledge in fighting the Alzheimer's disease. But all the modern findings suggest that actually the impairment or decrease in proper TREM2 functioning contributing significantly to the development of Alzheimer. In our work, we are trying to understand and harness one particular valuable commodity, extracellular vesicles and exosomes in particular. Exosomes are very small vesicles that can get secreted by cells either upon changes in environment or as a part of their natural process, and can play actually critical role in many brain functions. While exosomes can be secreted by all cells in our body, in our work we particularly focused on glia exosomes. Those tiny vesicles that are coming out of those cells are loaded with myriads of proteins, lipids, RNAs, and can play a crucial role in many processes, from controlling neuronal connections to neurotoxicity and neurogenesis, as well as can have pro and anti-inflammatory effects, which actually are very important in the context of Alzheimer's disease. Also, exosomes are considered novel breakthrough therapeutic modality for many diseases. While exosomes can have some obvious roles for Alzheimer's disease, exosomes are important for several reasons. And one in particular, that they mediate release of insulin-degrading enzyme, an enzyme that is actually critical for degradation of beta amyloid. Also, they are important for intercellular communications and distribution of key molecules, crucial for regulation of inflammation and neuronal survival. There are some conflicting results present in the literature, but we're trying to figure this out. There are two ways. The exosomes can be good as well as bad for neurons and other cells. There are indications that exosomes can be as anti-inflammatory as well as pro-inflammatory. They can improve neuronal survival. At the same time, there is data that actually indicates that they can damage neurons as well as decrease their plasticity and ability to survive. So there are two ways we can actually harness exosomes in fighting the Alzheimer's disease. And interestingly, there are ways for us to control release of exosomes as well. First, we can take internal cellular mechanisms like a case of serotonin pathway. It has been shown and it has been known for a long time that actually increased level of serotonin reduces risk of getting Alzheimer's disease. Either is by taking antidepressive drugs that actually increase levels of serotonin. You can actually boost production of exosomes from glia cells. And this boost in production of exosomes results in release of insulin-degrading enzyme that actually can promote degradation and reduction in amyloid plaques load. At the same time, those exosomes can reduce inflammation in the brain, which is also actually very beneficial for cell survival. Similar story we see with statins. Well, many of you know what the statins are and why many people actually are taking them. They are also known to reduce chances of dementia, and there is actually a very robust literature on this. In similarity to serotonin, statins actually increase secretion of exosomes, which in turn can reduce levels of amyloid in the brain. And we have shown with others that taking statins can significantly reduce the amyloid, can increase the amyloid degradation. While both serotonin, which can be found inside our bodies and we actually produce it ourselves, or statins that can be taken as medication, show promise in reducing risk of getting a disease, and one can boost exosome secretion in the brain. However, both substances can affect many other processes in our bodies. And in our work, we're actually trying to look for new and more specific modulators of exosome release, so where we can specifically target those exosomes that we consider be beneficial for neurons in Alzheimer's disease, and which in turn would improve neuronal survival and functioning without influencing other processes. There is, of course, another way of doing this. And since exosomes showed high promise in degradation of amyloid and have other extremely beneficial properties like reducing inflammation and promoting neuronal survival, one can actually produce exosomes on a high scale as a therapeutic modality. So basically we can use exosomes as a drug. And for this, we have partnered with the French company evolv to explore their unique technology that can help us actually to scale up production of exosomes that we are currently established in our laboratory. But the amount as well as the quality of exosomes is simply not good enough to transfer this to human studies. And hopefully with combining both approaches where we can target particular pathways to promote release of good exosomes from glia cells, as well as harnessing technology for mass scale production, we can come up with a solution where exosomes can be used as a therapeutic modality for Alzheimer patients, either in case of boosting their own production or using exosomes as an external drug that can be given to patients. As you can see, glia role in Alzheimer's disease is a very complex, and we have potential to harness the power of glia cells to fight Alzheimer's disease. However, we still have a long way to go because of complexity of glia's function, as well as big variability in glia's populations. And with this, I would like to conclude my presentation. I would like to pass to my colleague Jon from Exeter. Thank you.
Hi there. Thank you, Konstantin, and hello, everyone. I'll just take a moment here just to share my slides with you. And so hopefully everyone can see my slides there, and I'll just bring up my laser pointer here as well. So in this second talk in our session today, I'm going to touch on a few of the concepts that Konstantin has mentioned there, but what I really want to also focus on is taking a look at the role that interactions between glia and neurons play in our evolving understanding of the importance of this process in the maintenance of normal brain health and how this goes wrong in dementia causing illnesses such as Alzheimer's disease. And I should mention that I'm Jon Witton, for anyone not aware, and I'm based at the Institute of Biomedical and Clinical Science at the University of Exeter. So, as Konstantin mentioned in the last talk there are two broad categories of cell within the brain. So first we have our neurons, otherwise known as nerve cells. And these cells are primarily involved in the communication and processing of information within the brain. But neurons can't do this alone, and they require the support of an additional category of cells called glia cells. And two types that I want to draw your attention to in this talk are microglia cells and astrocytes. There are some other types of glia cells, but these are the ones I want to focus on here. And these cartoon schematics give a rough indication of what these different cell types look like in the brain. And so neurons and glia cells come together to form functional neural circuits within our brains. So for example, here we have 3 neurons that are connected together. And so in this schematic, information would be passed from this neuron here and this neuron here to this cell. And these cells are then surrounded by astrocytes, which play a key role in maintaining the brain environment and providing nutrients to these neurons, clearing waste products, and also we're starting to understand how these cells also play an active role in facilitating communication between neurons as well. And then we have our microglia cells. And you can think of microglia cells as sentinels within the brain. These are the brain's primary immune cells, and they survey the brain environment looking for toxic changes or harmful changes and trying to address these. And so this is the kind of organization that we might expect in a neural circuit in a healthy brain. And just to illustrate this, what I'm showing you here is a section of tissue taken from a brain that's been labeled with fluorescent stains for these different cell types. And so what you can see here in this brain tissue is, here we have a couple of neurons, which are labeled in red. And these are surrounded by these star-like astrocyte cells, which in this case are labeled green. And then we close by, surveying the brain environment, we have our microglia cells, in this case labeled in blue. Now, that's the scenario we can think about in the healthy brain. But one of the features of the brains of many people that have dementia is the buildup of toxic aggregates of protein, and what we might generally refer to as protein-based pathologies. And so if we consider Alzheimer's disease, which is the most common cause of dementia, accounting for around 70% of cases, there are two main types of protein pathology that we observe. First, we see amyloid plaques, which form outside of cells. And then we see tau tangles, which form inside cells. And these pathologies emerge across an extended period of time, often years or decades, and often prior to the emergence of the symptoms of the disease. And there's a sequence to this process. So if we consider the amyloid plaques, which are one of the first pathologies to emerge in Alzheimer's disease, what we first see is the deposition of single monomers of amyloid beta protein. So these are single proteins that are released from individual cells. And under normal conditions, this isn't problematic. This isn't a pathological process. But what happens is that these proteins can become misfolded, misshapen, essentially. And this causes them to start to clump together. And so what we first see are these proteins rather clumping together to form these small clusters that we call oligomers, then these larger structures called fibrils, and then finally these plaques that we can see down the microscope. And so what you're looking at here is a section of brain tissue from an Alzheimer's patient where the plaques have been labeled with this brown stain. And so if we map this pathological process onto our neuronal circuit, what we first see is the deposition of our monomers of amyloid peptide. These then clump together to form oligomers, then fibrils, and then these large protein-based structures called amyloid plaques. And I mentioned earlier on that glia cells regulate the immune responses within the brain. And so these glia cells recognize this process of this development of pathology and try to respond to it. And so the development of this pathology causes the properties of glia cells within the brain to change. And so a couple of things that we see are illustrated here on this slide. So if we first consider our astrocytes, one of the things that we see in these cells is that they take on what we call a hypertrophic appearance. The processes or arms that come out of the cell increase in number, and they become thicker, and the cell overall becomes more massive. Conversely, what we see with our microglia cells is that these lose their processes or arms and take on this more amoeboid or blob-like shape. And in this configuration, one thing that these cells do is to release inflammatory molecules into the brain to drive and coordinate an immune response, much like you might get inflammation in another part of your body when it becomes damaged. Now, over a short period of time, this isn't problematic, but over years and decades, as is the case of the development of dementia, this can be in itself pathological and damaging to the brain. Another thing that I want to, I particularly want to draw your attention to these microglia cells, because A process that these cells undertake when in this amoeboid or blob-like morphology or form is phagocytosis. And so what these cells do is essentially go around and engulf or eat these pathological protein-based deposits. And this is part of the coordinated immune response in the brain to try and clear these pathologies away. What microglia also do is to engulf or eat away connections that form between nerve cells, and these connections are called synapses. And when this process is properly coordinated and regulated, this is part of maintaining normal and healthy levels of activity within the brain. But what we're increasingly starting to see is that this process becomes dysregulated in dementia, such that synapses are being excessively pruned away. So those sort of connections between nerve cells are being pruned away when they shouldn't be. And indeed, one of the things that genetic work that's taken place over the past decade or so is starting to show us is that a number of genes that increase the risk of people developing Alzheimer's disease mess up with these processes, either by decreasing the ability of our microglia cells to chomp up these pathological protein-based aggregates, or increasing the prevalence with which our microglia cells chomp away at the synaptic connections, thereby disrupting the function of neural circuits and the information flow through those circuits. So I now want to just segue away from this slightly to talk about my interest in this area, because this is something that I've become particularly interested in over the last couple of years or so. And by training, I'm a neurophysiologist, so I'm particularly interested in how neurons within the brain work and how they process information. And one of the things that we see is that when lots of neurons or nerve cells within the brain communicate with one another, it generates waves of electrical activity, and these are colloquially referred to as brain waves. And we can record this electrical activity using electrodes. And we can do this in people by placing electrodes on the scalp, and you may indeed have seen this in the past. So here we have an individual with this rather fetching looking swim cap type device on their head. And these individual points here are electrodes that are recording electrical signals from the scalp. And so what you're looking at here on the right-hand side of the screen are waves of electrical activity being recorded from these electrodes on this individual's head. And in addition to doing this in people, we can also do this in models, in animal models. So for example, mice that have been genetically engineered to develop the kinds of pathologies that we observe in dementia patients. And so one of the things that we and other researchers have shown is that the kinds of pathologies that we see in Alzheimer's disease disrupt particular types of brain waves. And one important type of brain wave that we see disrupted, that's particularly important in cognitive functions such as reasoning, learning, and memory, is the gamma oscillation. Our work showed us that this seems to be disrupted by Alzheimer's pathologies. Now, it's been known for a number of years that neurons and glial cells release molecules that allow them to communicate with one another. But around 5 years ago, this paper came out in the journal Nature, which prompted the field to really start to rethink the mechanisms or processes that might underlie this communication between neurons and glia cells. And essentially what this paper showed was that in an Alzheimer's mouse model, if they used artificial methods to boost the levels of these waves of electrical activity, these gamma oscillations in their brain. What they also showed was that the properties of their microglia cells, remember these are the sentinels of their brain that coordinate and direct these immune responses, they also changed. So the cells went from having this star-like resting appearance to this more amoeboid, Pac-Man-like appearance in which they can go around and chomp up pathological aggregates. And remarkably, what they also observed in this paper was that when they boosted gamma oscillations and caused the microglia to take on this, this particular form, the overall amount of amyloid plaques that formed within the brains of these model mice decreased quite dramatically. And so this suggests that leveraging communication between neurons and glia cells and restoring normal patterns of activity or normal patterns of communication between these cell types could hold a key to treating Alzheimer's disease. And so one way that this group has taken, or one approach that this group has taken to do this is by using sensory stimulation in the form of light and sound to try and boost gamma oscillations in the brains of people with dementia. And so here we can see a member of this original research team wearing a device that provides light and sound stimulation to boost these gamma waves within the brain. But one thing that particularly interests me and my group is that we don't have a good handle on the mechanisms that underlie this process. And perhaps if we understand the molecular mechanisms that mediate this process, we could also target it with drugs to boost this process within the brains of people with dementia and thereby take an approach to treating the disease. Now, one of the difficulties that we have in trying to understand the mechanisms that underlie this process is that it's very difficult to get access to the brains of either people with dementia or even animal models. And so one of the things that we've been doing over the past year and a half or so is to develop a model in which we can study the molecular mechanisms that underlie this communication between neurons and glia cells mediated by these rhythmic patterns of brain activity. And we do this using slices of brain tissue. And we can place electrodes in this brain tissue, and we can also image this brain tissue and then use drugs to try and change the patterns of activity or block certain processes to understand how this works. And so one of the things that we observe in our model is that when there's an absence of this rhythmic brain activity, which is shown here, so this is a trace or a recording of electrical activity in this slice of brain tissue, what we see is that our microglia cells have this star-like resting appearance. So they're not in this sort of inflammatory state that can facilitate the clearance of protein-based pathologies. But what happens when we instate these rhythmic patterns of brain activity, these gamma oscillations in our brain slice, and so that's illustrated by this repeated pattern of oscillatory activity in our electrical trace. What we see is that our microglia change their appearance, so they take on this amoeboid-like morphology, and it therefore puts them in a state in which they're better able to engulf pathological protein-based deposits. And so what we're trying to do is to use this model to try and understand the particular signaling pathways that underlie this process, in the hope that by understanding these molecular signals, we could then target them with drugs. And so just to summarize, I've mentioned that glial cells play a crucial role in maintaining brain health and function, and that pathologies that are linked to dementia alter the properties of glial cells in the brain. And so perhaps by regulating the function of glial cells, and particularly by looking at how neurons in glia interact and reinstating normal patterns of neuron-glia communication, we might be able to identify routes to treating conditions that cause dementia, such as Alzheimer's disease. And one way that we've taken to doing this is trying to develop a model to understand how rhythmic patterns of brain activity, these gamma oscillations, regulate the function of microglia cells. And so with that, I'd just like to finish up. There's a few people I'd like to thank. First of all, colleagues at the University of Exeter, particularly Megan Ellie, who's been driving this work, and my collaborator, John Brown, and then also the funders, without whom we wouldn't be able to do this work. And so thank you very much. And I'll pass back over to Katy.
Thanks, Jon. And we'll just wait for Konstantin to join us back on screen, and we'll move into our Q&A sessions. That was really fascinating to hear all about microglia and astrocytes, these brain support cells and what they do in the brain. So if anybody listening has any questions, do pop them in the Q&A, and we'll put them to our speakers. So we've had one question come through already, and Julie is asking, and I think, Jon, this is in relation to what you were talking about with the gamma oscillations. She's asked, at what age could problems begin? So do we have any knowledge on, these brain waves are changing, but do we know when that might begin if somebody's developing dementia?
Yeah, so the problem is that the development of these pathologies are highly variable between individuals. And so it's very difficult to put a specific age bracket on when these changes might emerge. But if we operate on the premise that the development of these protein-based pathologies are what drive disruptions in these oscillations or these patterns of brain activity, and that they can emerge early, so before the onset of clinical symptoms, then what we can say is that we're likely to see changes in brain activity before the onset of the clinical symptoms of the disease. And so that's something that certain individuals around the world are working on, is using these electrical patterns of brain activity to develop diagnostic markers of the disease. Now, of course, the main risk factor for a number of dementia-causing illnesses, such as Alzheimer's disease, is age. And so this is likely to emerge later in life. But in terms of putting a particular sort of narrow age bracket on it, I don't think that would be a reasonable thing to do.
I guess because, studies over recent decades have indicated that proteins build up in the brain maybe 10, 20 years. So it's quite a big window, isn't it, that we're looking at, that some of these changes take place. So yeah, it's probably one of those earlier features. Konstantin, I had a question for you. So you were talking about exomes, and you're talking about how actually they seem to be able to be both good and bad. They seem to modulate activity and change activity in both directions. So is that because of what's contained within them? So they're like little packages, aren't they? And they signal what to do. So is it what's within them? And do you know what's within them that means that they might push activity up or down? Is that something that you can talk about at all?
Yes, in fact, actually, we can talk about exosomes as tiny parcels being delivered from one cell to another. And studies with proteomics especially, they actually identify that there are different pools of exosomes, and those pools, depending on the state of glia cell, can vary. So the proportion of, let's call them good exosomes, can be decreased, and let's say bad exosomes with pro-inflammatory factors can actually go up in let's say in dementia in general, or in other conditions as well. So there is definitely this kind of a mix of good and bad exosomes, and it would be great actually if we can find the way to steer the ratio towards good and reduce the amount of bad ones.
Fantastic, thank you. So we've had another question come through, and Joseph is asking, would AI be of use in identifying problems with these cells early on, or has this been started already? I wonder if either of you want to comment about how AI might be helping to pick up these changes that you see in your work.
Yeah, so I mean, one thing that we can say is that AI is becoming incredibly valuable across a whole range of medical applications for the early diagnosis of disease. So, for example, it's possible to develop algorithms that are able to detect certain changes in biomarkers of disease far more effectively than even clinicians are able to do. And so, yeah, artificial intelligence algorithms can be particularly valuable in terms of being able to analyze the sort of screens that we're able to do in patients, whether it's like we were talking about earlier, recording electrical activity of the brain or scanning the brain, for example, for these pathological changes, and detecting early changes that are going on in the brain that might not necessarily be so easily detected by clinicians or even by researchers who are looking at the data, but things that are very subtle can often be detected by these algorithms. So yeah, it's got a particularly important role to play as we go forward, I would say, in diagnosis.
Yeah, no, we've got a really large project that's trying to do that. So our Alzheimer's Research UK have a really large initiative called the Early Detection of Neurodegeneration, or EDEN for short. And that is trying to use a lot of this insight as to the early signs that we might be able to pick up, such as these brainwave changes or changes in behavior and lots of different factors, and then bring it all together using AI. So maybe we should plan to do a lab notes on that topic at some point in the future if people are interested in hearing.
Like you say, Katie, there, I think what's becoming increasingly clear as well is that taking a multimodal approach to trying to detect the disease is going to become standard form. So, for example, it's unlikely to be a case where there's a single early stage test that definitively demonstrates whether or not an individual has dementia. It's the multimodal approach. And again, using AI algorithms to be able to integrate information across different diagnostic tests is also a way that sort of high level computing is useful in this area.
I guess, and that speaks to the fact that Alzheimer's and all these different types of dementia are really complex. So there's not probably one single cause. There's lots of different changes happening that come together to create these diseases. And so, pulling information from lots of different factors is important. We've had another question come through, and I wonder if, I know you briefly mentioned a gene that's been linked with particularly microglia, but Vern is asking, are there any hereditary or family links noticed? So I think this is more of a general question about the link between genes and Alzheimer's risk. I didn't know if either of you wanted to talk any more about what we know about that, and maybe about this this gene that is linked with glia.
Well, there is definitely a very strong link between TREM2 mutations and developing Alzheimer. And actually, if we look carefully at the data, the mutations in TREM2 can be as strong as mutations in ApoE in predisposition to getting a disease. If we will talk about exosomes in particular, I am not aware of any genes that actually related to exosomes, either as the gene that is involved in regulation of the secretion or production of exosomes, that is actually linked to any disease so far. So there should be, taking how important exosomes from glia cells are for everything basically in the brain, for bad and for good, there should be definitely some links between exosomes and genes and so certain conditions. But I think we're still just starting to understand. In fact, about exosomes, it's actually very interesting because the original research, actually discovery of exosomes, is 39 years old. But if you will look at state of research, it's basically just recent years we started to understand how important and even crucial they are for the brain health, let's say, in general. So we just basically just starting this journey, though actually the first brick was laid out 39 years ago.
So I think one thing that I'd say on that is, I started working on dementia around about 10 years ago, I suppose. And at around that time, the study of glia in the context of dementia was pretty niche. It wasn't particularly fashionable. And one of the things that's really changed that is the development of genetic technology over that period of the last decade. And what's that done is identify a number of risk genes that are related to the function of glia cells. So these are not genes that will definitively cause any individual to develop dementia, but they heighten individuals' risk of developing dementia. So for example, the TREM2 gene that Konstantin was talking about, that interferes with the ability of microglia to take on that Pac-Man type role and chomp up these pathological proteins. Some of the other risk genes increase the frequency with which microglia seem to indiscriminately prune away synapses, and so disrupt the ability of information to flow through neural networks in the brain.
Fab, thank you. Yeah, I think certainly when I was studying microglia in my undergraduate degree over 10 years ago, that we weren't thinking of them in terms of diseases like this. So yeah, these genetic discoveries are huge. Yeah,
and I think that's caused such an explosion over the past decade in terms of the overall interest and focus on the role of glia. But we're somewhat lagging behind in terms of our understanding of both the role of these cells in general and how they function in the normal healthy brain, but then also the kind of roles that they play in pathological states such as dementia.
Can either of you remember off the top of your head how many people carry one of these TREM2 mutations? I think it's really important to note that they are relatively rare. They're risk genes, so it doesn't mean if you have one of these variants, you're definitely going to get Alzheimer's disease, but it does is meant to increase your risk. But I think it's a relatively rare variant, isn't it? So it's like less than 1 in 50, maybe.
TREM2 is really rare, so this is not so common. But nevertheless, the effect of the gene mutation actually on chances getting the disease are very, very strong. So it's a strong mutations.
Wonderful. Changing tack slightly, there's a question come through that I'm quite interested to ask you. So Joseph has asked, have we explored environmental factors or changes that affect the health of our glial cells? So organizations are into taking care of the planet in relation to our health. So is this something we know about, or is it too early days, do you think?
Yeah, so just to sort of generally speak to that, it's almost, well, it's the case that there's an interaction between an environment and then also, if you like, genetic risk predisposition. So for example, one of the things that we know is that systemic inflammation, so this is inflammation within the rest of the body, increases one's risk for developing a dementia-causing illness. And also then conditions which drive inflammation in the rest of the body. So for example, diabetes being one, increase the risk of developing a dementia-causing disease. And one of the reasons for this is that processes that drive inflammation in the rest of the body also drive inflammation within the brain. So this is one of the reasons that the current best recommendation of in terms of how to reduce dementia risk is to be as healthy as possible. It's rather sort of unsexy advice, but having a low weight, being active, being fit, having a low blood pressure, all these things decrease one's risk of developing dementia, and inflammation's one of the parts to that process.
Brilliant, thank you. We've had a bit of a technical question come through for Konstantin, which I think we might try and answer offline, because I think it's probably going to get in too much technical detail for this sort of forum. But just as a final question, as I would say relatively young, eager researchers, what's the biggest or most exciting area of research that you see at the moment, and kind of where you get the most hope from when you're doing your research? So Konstantin, I'm going to put you on the spot and see what's the most exciting area of research for you at the moment?
Well, it is definitely exosome research, because it's just booming. And as I said, so there are two different big directions. It's either to modulate the internal processes for exosomes, or whether use exosomes as a drug. And all the data that actually comes out now, it shows that exosomes can be really big deal breaker in those things. So they can be really boosting protection properties of other drugs. Actually, they can support other drugs as well. So that for me is definitely glia plus exosomes is the next frontier.
And to you, Jon, your most exciting area?
Well, so what I'd say is in terms of giving most hope, one of the things that I'm sort of most excited about and hopeful about is the overall level of coordination that there now seems to be in the dementia research landscape. So again, roughly if we go back 10 years ago, we had pockets of dementia researchers within the UK and throughout the world operating, if you like, in sort of their own silos. And what we're now seeing is that research is becoming far more integrated. There's far more active collaborations. We have organizations which are coordinating large-scale programs to try and direct funding into areas where big research can be done, for example, and in terms of generating big data sets that can be shared across labs, coordinated efforts to take a sort of multidisciplinary approach to looking at the illnesses. So that might be, for example, translating findings all the way through from cells in a dish through to animal models, through to patients, and then drug discovery. So I think what we're starting to see is a far more organized and coordinated approach to understanding dementia. And I think this is going to yield much more, much quicker developments, but also sort of more blue skies developments in terms of our understanding of dementia over the next sort of 5 to 10 years.
Brilliant. Well, that's such a positive note to end on. So in the interest of time, I'm going to say thank you to Konstantin and Jon for your talks and for answering our questions, and just hold on with our audience a second longer because we've got a poll and some final details for you. But thank you, Jon and Konstantin. It's been great.
Thank you.
So before everybody drops off the event right now, we've just got a couple of poll questions we'd love you to answer to give us some insight into how you found today. So we're interested in finding out whether you found the content of the talks about right or a bit too technical or simple, whether you'd recommend these events to your friends and family, and whether you think you've learned something today. So just pause a moment while you can answer those. It's always nerve-wracking as I sit here and watch these come through, but I think it's looking pretty positive, which is great. So I think, yeah, pretty much everybody has had a pop their answers in, so we'll share them back with you. So content was about right, which is great, and people, you would recommend these events to your friends and family, and you think you have learned something today. So that's absolutely fantastic to hear. Thank you very much. And we'll be sending a more detailed feedback survey to you later today, and the video recording will come later in the week. Please do complete the survey. I would appreciate any feedback you can give us about the event, as we're also planning our 2022 series now. So we're seeking any input you have about topics or what you'd like to hear about next year. And our next event in this current Lab Notes series is on Thursday, the 7th of October at 1 p.m, where we'll hear from researchers in our Northwest network who will share their research into vascular dementia. They'll talk about things like the link between high blood pressure and dementia, and also how the protective layer of the brain can be disrupted in this type of dementia. And you can find out more about this event and sign up on our website to join us then. Also want to give just another little reminder of our Dementia Attitudes Monitor findings that I talked about earlier and that we've released today. Do check out the website and the full report if that is of interest to you. And if there are any questions that we weren't able to answer, or you're seeking some more kind of personalised support around anything, please do get in contact with our Dementia Research info line. They can answer questions about dementia, signpost to other services and information, and also sign you up to research studies if you're interested in taking part. So as we close, I just want to say a huge thank you for joining us today. I hope you really enjoyed it and found it useful, and that you will come along again for another event in our series. Thank you and goodbye.